N. So this is the second semester, chapter on metabolism. I just called this intermediate metabolism. And so in the first place, we saw that the end product of glycolysis was pyruvate. So what can happen to pyruvate? Well, most pyruvate enters the citric acid cycle. It's got to be turned into acetyl coenzyme A. So here's that right here. So pyruvate gets converted into acetyl coenzyme A. You saw the structure of coenzyme A. Here is the acetyl group. That's 2 carbons, right? Similar to acetic acid. And during that process, this 3 carbons gets turned into 2 carbons. So this is carboxylic acid that gets cut off, decarboxylated, and we get CO2. So here CO2 is produced before we get through the Krebs cycle. And if we look, we also take NAD and turn it into NADH. But because this happens in the mitochondria, NADH goes right into the electron transport system and it gives us 3 ATPs. And this enters the citric acid cycle. But a couple other things can happen. So pyruvate can be carboxylated into oxaloacetate. It's like, well, why would you do that? Because you'll see that oxaloacetate is absolutely necessary to keep the citric acid cycle going. Oxaloacetate can also be used in gluconeogenesis. We'll look at that as well. So here's something that can go both ways. Pyruvate can be turned into lactate, and lactate can be converted back into pyruvate. For the most part, you convert pyruvate into lactate for the single purpose of regenerating your NAD. That's the reason why you do it. You go back to glycolysis. If you don't have NAD, glycolysis cannot continue. You need glycolysis to continue because that's how you get ATP made without the presence of oxygen. But you need NAD to do that. So if you had glycolysis going and you had converted all of your NAD into NADH, but then the mitochondria can't oxidize that NADH, you don't have any more NAD. And you're stuck and you're going to die. That's the only reason why we make lactic acid. So lactic acid, most time I would say glucose goes into the Cori cycle to produce more glucose. So it's returned into glucose. So what about this direction over here? Pyruvate can again be decarboxylated, but this time simply into the aldehyde, acetaldehyde. That can then be reduced into ethanol by yeast. What happens in bacteria, there's another decarboxylation and you end up getting methanol. Methanol is toxic. Ethanol is also toxic, but we can tolerate it. That's what we get from, intoxicating liquors, right? So why would a yeast or a bacteria do that? The only reason is it's going to do it only when there's no oxygen. And again, for the same reason we make lactate, they need to regenerate their NAD as well. Because in the lack of oxygen, they would simply have no way to make ATP from glucose. So I use all those words there, I would say. And then one thing that's kind of interesting is the Warburg effect. They found that tumors produce up to about 85% of their pyruvate is converted into lactate, even in the presence of oxygen. So it turns out that these tumors are deficient in an enzyme called histone deacetylase, and that enzyme is necessary to remodel the histones so that you can actually get to the gene. So that gene, the gene for this enzyme, is also thought to be a tumor suppressor. So it's an interesting effect there. So here's the Cori cycle. So you produce most of your lactate in the muscle tissue. That's when you do anaerobic muscular activity. Goes through the blood. And in the liver, especially, lactate is converted back through pyruvate. Pyruvate can be converted into glucose by gluconeogenesis, and glucose goes back in the blood, and you get this cycle. That's why it's called the Cori cycle. So now we'll look at gluconeogenesis. So look at this. I tried to put a whole bunch of stuff onto this picture right here. So here's glycolysis. So glycolysis is in red, starting from glucose going down to pyruvate. Here's gluconeogenesis, making new glucose from pyruvate. Of course, lactate can be converted into pyruvate. Some amino acids can enter glycolysis. Those are called glucogenic amino acids. Anyway, a couple of things here. So we had some control points, right, in glycolysis. And then down here's another one over here. Okay. But remember, this is a very high energy molecule right here. When PEP goes to pyruvate, we make an ATP out of substrate level phosphorylation. So this is a very large energy drop right here. So to get from pyruvate back into PEP, we have to make another step. Lowering the energy of activation to get from here to here, we have to go through a second step. So there's a second enzyme. This happens to be a control point. So the control point here is with pyruvate carboxylase. This is a allosteric inhibitor, allosteric enzyme. It's inhibited by ADP, which means what? If you have a whole bunch of ADP, that means that the cell doesn't have a lot of energy. So if you don't have a lot of energy, why would you be trying to synthesize glucose? But if you do have a lot of energy, you're going to have a bunch of ATP, which would activate pyruvate carboxylase, and a bunch of acetyl coenzyme A, which is also indicative that there's a bunch of energy around. Maybe take some of that energy and use it to make new glucose. So notice that between here and here are different enzymes. What about here? That's the same enzyme, same enzyme, same enzyme. My God, these are all the same enzymes. What does that mean? That means that glycolysis and gluconeogenesis occur in the same compartment of the cell, both in the cytoplasm. So it's absolutely logical to use the same enzymes as much as you can. But you have to be able to have some way to control it, and that is with pyruvate kinase, right? Pyruvate carboxylase over here, different enzymes. Same up here, different enzymes. They're controlled by different things. So fructose 1,6-diphosphatase is turned on by citrate. If you got citrate built up, that means that you got a lot of energy hanging around. Yeah, we want to make glucose now. But all these other ones indicate that we don't have a lot of energy. So let's turn this enzyme off. But these are the same things that turn this enzyme off are the same things that turn this one on. So remember, it doesn't make any sense to at the same time oxidize glucose to get energy and at the same time take pyruvate and synthesize glucose. Makes no sense. So that's why you have to have certain enzymes that are allosteric control points. So I went through the different steps over here, the different control points, okay. Let's see. Notice that glycerol can be converted into dihydroxyacetone phosphate in interglycolysis. So here is glycerol being phosphorylated and then being phosphorylated. And here is basically where lactate can be produced. Okay. So pyruvate can either go through gluconeogenesis through the Cori cycle, be converted into acetyl coenzyme A, or get converted into oxaloacetate. So here's an overall viewpoint. And so here's some indicators of high cellular energy. If a cell has a lot of energy, you have large amounts of ATP, GTP, reducing agents, citrate, acetyl coenzyme A. Low cellular energy is going to mean you have more of this stuff, right? It makes perfect sense. So then I went through the different control points in quite a bit of detail. And so what is the role of fructose 2,6-diphosphate? Well, it inhibits gluconeogenesis at control point 1. It's an allosteric inhibitor. Okay, but why? Because this is a way that you can control the amount of fructose 2,6-diphosphate by using glucagon and insulin. So in other words, it's a way for your pancreas to control gluconeogenesis versus glycolysis. So how does glucagon do this? Okay. So you either get glucagon or adrenaline, and you end up producing a second messenger, cyclic AMP. So that goes to protein kinase A, and now you get less fructose 2,6-diphosphate, and that turns off glycolysis. This is what glucagon or adrenaline does, turns off glycolysis, turns on gluconeogenesis. And how does it do that? Well, here's these two enzymes right here. What protein kinase A does is simply goes around putting phosphates on whatever proteins are in that particular cell. So in a liver cell, for example, you have this enzyme complex. So here's phosphofructokinase 2, and here's fructose dephosphorylase 2. When you put a phosphate on this complex, you turn off this enzyme, which turns off glycolysis, and you turn on this enzyme, which turns on gluconeogenesis. So what does glucagon do? Glucagon tries to ensure that you increase blood glucose so that you have enough energy for your body to do a fight or flight response, or do something physically challenging where you need a lot of energy. And what about insulin? How does insulin do this? Well, it's released by the pancreas. Then it turns on protein kinase B. So this is a protein kinase A, this is a protein kinase B. And what does it do? It turns on phosphoprotein phosphorylase. What does it do? It removes a phosphate. So the protein kinase put the phosphate on, turned off this, turned on this. But when you activate a phosphorylase, it takes off the phosphate. That activates this enzyme and inhibits this enzyme. So you get more fructose 2,6-diphosphate. That goes on and it activates glycolysis, turns off gluconeogenesis, and it decreases blood glucose. Okay. If you look at the other two control points, so an activator, for example, insulin increases hyperglycemia. Inhibitor, glucagon, would cause hypoglycemia. So when it comes to glycogen synthesis, this is a different story. So if we go back over here, we looked at several ways to turn on this and at the same time turn off that, and vice versa. But the common denominator is glucose. If the cell needs energy, you burn glucose. If the cell already has enough energy, what are you going to do with that energy? Well, you need to store it because you don't even know when you're going to be able to eat the next time. So storage means you produce glucose, right? So if glucose is high, how do you store glucose? You store it as glycogen. So notice the two words, glycogenesis and glycogenolysis. Okay, so this is simply taking glucose and adding it to existing glycogen, or breaking glycogen down into glucose for use in the blood. So let's look at the synthesis of glycogen. There would be a problem. Here's a glucose 1-phosphate, which you would get from the first step of glycolysis, right? That's cellular sugar, right? And you go to put it on the glycogen. So every time you do that, you add a glucose 1-phosphate to glycogen, you produce a phosphate. Now that phosphate is a problem because high levels of phosphate, just simply using Le Chatelier's principle, is going to push the reaction this way. So removing a phosphate every time is a problem. So they had to get a workaround. When I say they, I mean, the people who designed the cells. The fix is, instead of using glucose 1-phosphate, the cell uses glucose UDP. Okay? So what the heck is that? UDP is like uracil diphosphate. Okay? So why would it choose that? Well, who knows? But here's the fix. So when you add this to glycogen, you don't get phosphate. So UDP can be used for other things, such as biosynthesis. Okay? Biosynthesis of what? RNA, for example. So then you have to have an enzyme that takes glucose 1-phosphate and UDP and makes glucose UDP. So if you look at this fix right here, you end up giving off pyrophosphate. So again, pyrophosphate buildup would be Le Chatelier's interference, but that gets broken down into phosphate. So phosphate now is not involved in this particular equilibrium. It's not involved in this one anymore. So you can just go into this. You just have to make sure that you keep making RNA, but that's not a problem because the cell makes RNA all the time. If the cell can't make RNA, then the cell dies pretty quick. So you look at this and you find out that an ATP was used to convert blood glucose into cellular glucose. It takes the equivalent of 3 ATPs to synthesize the glucose UDP from blood glucose and to add that to glycogen. So this is one reason why the cells cannot be 100% efficient. But the reason really is they're trying to circumvent the effect of Le Chatelier's principle. So any questions so far? So what about glycogenolysis, the release of glucose? So the alpha-1,4 bonds in glycogen are cleaved using inorganic phosphate. So remember, this is the chemical reaction and the enzyme is just a catalyst, right? So what we're really doing, we're taking phosphate and we're jamming that in here between the alpha-1,4 bonds, and we're replacing that bond with one of the glucoses with a phosphate. And again, we get glucose 1-phosphate. So again, if you're going to break it down even further, if there's like a severe drain on your glycogen, then you would also need debranching enzymes. Okay, so alpha-1,6 glucosidase, right? Okay. So now we have another set of 2 processes. This one makes glycogen. This one breaks it down. How do we ensure we're not making glycogen at the same time we're breaking glycogen down? So you have to have control. So it's basically controlled with using hormones. Okay. Again, insulin and glycogen. Okay. So glycogen going into glucose is catalyzed by glycogen phosphorylase. Phosphorylation activates that enzyme. Glucose turning into glycogen is catalyzed by glycogen synthetase. When you phosphorylate it, then that turns off that enzyme. Make sense? So your pancreas secretes insulin and glucagon. Your pancreas controls blood sugar, but it also controls the storage of sugar in glycogen and the breakdown of glycogen. So here's a rather complex way of doing this. So here's insulin binding to the insulin receptors. That's a kinase. And what it does, if you remember, it puts an ATP on some protein. Okay, well, if you're a liver cell especially, one of the major proteins over here is going to be this kinase. What does the kinase do? You turn it on by putting the phosphate on it. So the kinase that's part of the insulin receptor activates another kinase, phosphotransferase, by phosphorylating it. So then protein phosphorylase takes glucose UDP and basically turns off the phosphorylase. Okay, so you turn off the phosphorylase. So glucagon binds to its receptor. It goes through a second messenger, right? Cyclic AMP, turns protein kinase on. It puts a phosphate on glycogen phosphorylase, And what does that do? It turns that enzyme on, and then it simply starts breaking down glycogen. Let's see. Oh yeah, pyruvate kinase. I have to carefully write him over here. So what about pyruvate kinase? Okay. So if you remember what pyruvate kinase does, where did it go? I didn't put the enzymes up here. Sorry, I thought I had done that. Okay. So again, pyruvate kinase is another allosteric enzyme. It's activated by insulin, inhibited by glucagon. Okay, so pretty much similar to what's going on right here. Okay. So what I wanted to do is I made this figure like the other day. I didn't have time to put it into the chapter. So I guess I'm going to call it a figure 1 maybe, I don't know. But I just wanted to give an overall view of the glucose metabolism. So here's the cell. Glucose outside the cell has to be transported into the cell by the glucose transporter. If you remember from chapter 31, the glucose transporter is not there until certain things happen. So glucose, this can't randomly go into a cell just because it's higher out here than it is over here. It's only under certain circumstances. Then glucose goes through glycolysis into pyruvate. During that time, you also make NADH. So the NADH gets carried into the mitochondria by another transport protein. And they go through the electron transport system, and the purpose is to make ATP. And pyruvate enters the mitochondria by a translocate protein, and then pyruvate dehydrogenase turns it into acetyl coenzyme A. And then it can enter the citric acid cycle, which turns it into carbon dioxide. And at the same time, you make NADH and FADH2, which go into the electron transport system to make ATP. So there's compartmentalizations, including that of the cell. So any questions on that?
Is that all happening inside the matrix of the mitochondria?
All this here?
Yes.
Yes. Yeah. Okay. So how does pyruvate get into the citric acid cycle? So pyruvate translocase imports pyruvate into the mitochondrial matrix and symport with hydrogen. So in other words, here's pyruvate. So it can easily go through the outer membrane, but as it comes into here, it's symport. So really it's the pH gradient that drives the import of pyruvate. So you know what I mean by symport, right? So pyruvate has to bind to the translocator protein, as does hydrogen. Then they both come in. But remember, we use energy to pump protons out to create that electrochemical gradient. So we're consuming some of the gradient just to get pyruvate in. So it's not exactly free to get it in. Similarly, the gradient is consumed when you import phosphate in exchanging ADP for ATP. So here's bringing phosphate in. Notice that it also is symport, consuming some of the gradient. Here's the ADP transporter. So ADP comes in at the same time ATP goes out. Okay? But all this stuff basically costs energy. Maybe not directly in the form of ATP, but yeah, you're consuming some of the electrical chemical gradient. So here's what the pyruvate dehydrogenase complex does. It's a very large enzyme. So it takes pyruvate. It has to be able to bind pyruvate, coenzyme A, and NAD. So when you make acetyl coenzyme A, there's a decarboxylation reaction. Not happening in the Krebs cycle. This is considered separate from the Krebs cycle. And then you also produce an NADH, but that's right there in the mitochondria, so it gets oxidized by the electron transport system to give you 3 ATPs. So what about reducing equivalents from glycolysis? How do you get those into the mitochondria? Notice that in this diagram right here, I didn't talk about that. The glycolysis is producing NADH out here, and we need to get that in here to start producing energy from it. Well, it's not exactly simple. In heart and liver, an NADH from the cytoplasm gives you about 3 ATPs. In muscle and nerve tissue, you get only about 2 ATPs because they're 2 different shuttle mechanisms. So here's basically what happens. So NADH is used to reduce oxaloacetate into malate. Notice that that NADH does not come in to the matrix, doesn't leave the cytoplasm. So malate comes in, and at the same time malate comes in, alpha-ketoglutarate goes out. It's a type of antiport. Well, anyway, now you've gotten malate into the matrix. It's then oxidized to oxaloacetate. Okay, now you produce your NADH, which goes to complex 1 and gets oxidized. So we didn't directly bring the NADH in. We reformed it in the matrix by bringing malate in. So now why is this called the malate aspartate shuttle? Well, now the malate is gone. It's in the form of oxaloacetate. So it gets converted into aspartic acid. Then aspartic acid can be translocated out, but at the same time, the glutamate is brought in. See what I'm getting at? So here's an antiport, and here's another antiport. Working in conjunction is necessary to get the NADH from the cytoplasm into the matrix. So what about the glutamate? I mean, what's the big deal with that and the alpha-ketoglutarate? Well, those are also used in the oxidative deamination of glutamic acid, and that also occurs in the matrix. So they're being repurposed here. So just to give you an idea of how intertwined all these things are, what if for some reason the cell was deficient in glutamate. What would that do to the efficiency of this shuttle right here? Yeah, it would kind of shut it down, wouldn't it? You know, when you start looking at the complexities of all these things, you start to realize that, wow, it sure is easy for something to go wrong when there's so many complexities in this, and just being alive, which really ultimately brings you to this point. It's really easy to die, isn't it? So many things can go wrong. Well, what about the glycerol phosphate shuttle? So we talked about muscle tissue having to stay warm, and the inefficiency can help give warmth to muscle tissues. What about nerve tissues? Psychologically, the way I think about nerve tissue is that they are the most specialized cells in the entire body. Stem cells being the least specialized, nerve cells being the most specialized. You have to maintain certain temperatures there and all kinds of other protections to help keep those nerve cells from dying, because you simply can't really regenerate them. Anyway, let's try to take a look at this shuttle system. So it's the glycerol phosphate shuttle. So here's glycerol phosphate right here. So here's the NADH from the cytoplasm. So it combines with dihydroxyacetone phosphate, which is right from glycolysis. Here's the enzyme that does that. It's a dehydrogenase from oxidoreductase, glyceraldehyde 3-phosphate dehydrogenase type 1. And so you reduce dihydroxyacetone phosphate into glycerol 3-phosphate. Notice that the NAD does not come in. So glycerol 3-phosphate can be shuttled in to the matrix. There is combined with FAD. So notice that this is called glycerol 3-phosphate dehydrogenase type 2. This is the mitochondrial version. This is the cytoplasmic versions. Those are isozymes. So then when you oxidize, see, first you reduce it, now you're oxidizing it, you produce FADH2. And the dihydroxyacetone phosphate can just go right out of the matrix through its shuttle, its transporter. And now you produce FADH2 that goes into the electron transport system at complex 2. So you skip the first proton pumping station, which is complex 1. This is why you only make 2 ATPs from an NADH in muscle and in nerve tissue. So anyway, those are all the peripheral things leading up to the citric acid cycle, which occurs in the matrix. So this Sir Hans Krebs was an amazing guy. They named the cycle after him because this was such a complex thing that he figured out. So the guy worked his tail off his whole life. I used to know one of his grandnephews, and I hate to say it, he passed away last year, but one of the laziest guys I've ever met in my life. So I guess the motivation that his great uncle had was not handed down to the rest of the family. So let's just get to the cycle. So here I had to draw this line. So here's the citric acid cycle. And here's where are we? Here's pyruvate. Here's pyruvate would come in. So here's pyruvate dehydrogenase complex, right? So that's where you're making the NADH and all that stuff, right? decarboxylation. And so the product of pyruvate dehydrogenase is acetyl coenzyme A. So that has 2 carbons, right? In the first step, acetyl-CoA with 2 carbons is combined with oxaloacetate. Now remember, one of the fates of pyruvate that we talked about in the very beginning of this chapter was it could be carboxylated back into oxaloacetate. And I said the reason why that needs to happen is because if you don't have oxaloacetate to combine with acetyl coenzyme A, the Krebs cycle stops. So notice that this enzyme called citrate synthetase has to be able to bind acetyl coenzyme A, oxaloacetate, and then it hydrolyzes off the coenzyme A, which can then be used again up here. And you get, through this condensation reaction, citric acid. So notice that citric acid is the name of the cycle. You got 1, 2, 3 carboxylic acid. It's a trioic acid. That's why it's also called the tricarboxylic acid cycle, the TCA cycle. So what happens to citric acid? And by the way, sometimes you see people say citrate, sometimes you hear them say citric acid. I prefer to say citric acid when it's in the matrix of the mitochondria, because I should say, well, I messed that up. I would prefer to use the word citrate when it's in the mitochondria, simply because when you're pumping protons out of the matrix, the pH in there is going to be alkaline. So more likely this is going to be more ionized, but nevertheless, it will still follow through. So what does the first enzyme do? So a conatase takes citric acid and turns it into cis-conatic acid. So what did it do? It took this hydroxy group and one of those hydrogens right there and put a double bond there. So the enzyme aconitase could be considered to be a lyase. Okay, well, what happens in the next step? The enzyme, what? Aconitase is there again. I know that gets confusing. So what did it do? It took the water and put it back into the molecule. The double bond is saturated again with water. And the product is called isocitrate. So if you combine these two steps right there, aconitase is really simply an isomerase. It took this alcohol group on the third position right here and moved it to the second position over here. So for some reason, this historically, the way it was discovered, it was in two different steps because they found both of these metabolites in there and had to explain it. So right now we didn't make any energy, did we? Simply brought in 2 carbons, combined it with 4, and now we got 6 carbons. So the first place where we make energy is with isocitrate dehydrogenase, and dehydrogenase are oxidoreductases. So this enzyme has to bind isocitrate and NAD, and it oxidizes it into oxalosuccinate. Okay. So that NADH now goes and makes 3 more ATPs. Okay. So right now, we've made 3 ATPs and we still have 6 carbons. So the next enzyme is oxaloacetate decarboxylase. And I'm sorry I keep using the ionized terms. So what does it do? Well, it's a decarboxylase. It mostly decarboxylates. So that's the first carbon that goes off. Remember, you brought in 2, and now 1 has gone off. So we have alpha-ketoglutarate, which you have seen in a couple of different places before. It's a metabolite in the citric acid cycle. It also has other purposes. I got 1, 2, 3, 4, 5 carbons. So then the next enzyme is a complex. That means it's a large enzyme, alpha-ketoglutarate dehydrogenase. So what does it do? It binds alpha-ketoglutarate, does another decarboxylation. So now there's going to be 4 carbons over here, right? And now you've gotten rid of the second carbon that you had brought in as acetyl coenzyme A. It also has to bind NAD, so you oxidize it, produce NADH, which gives you 3 more ATPs. And look, coenzyme A is brought back in. Kind of interesting. You brought coenzyme A with you, and then you took it off right away, and now you're putting another one back on. So the product of alpha-ketoglutarate dehydrogenase is succinyl coenzyme A. So this is succinic acid, but it's in a thioester right here with coenzyme A. So far, how much ATP did we make? 3 and 3 is 6. So the next step is succinyl coenzyme A synthetase. So what does it do? This enzyme binds succinyl coenzyme A. It has to bind phosphate. It has to bind GDP. And this is an extremely complex reaction. But what does it do? It takes GDP and phosphate and squeezes it together into making a molecule of GTP. So GTP is equivalent to ATP in terms of energy. It can later be converted into ATP. There's an enzyme that does that. But the immediate product is simply GTP. And because this was not made using the electron transport system, it was made on the surface of an enzyme. This is known as substrate level phosphorylation. So now we've made 3, 6, 7 ATPs, and we have still a molecule of succinic acid, or succinate. What is the next step? So succinate dehydrogenase, an oxidoreductase, has to be able to bind succinate and FAD, and it reduces FAD into FADH2, and you basically are oxidizing succinic acid into fumaric acid. But that FADH2 is equivalent to 2 ATPs because it enters electron transport system at complex 2. So now I've made 3 ATPs, 6 ATPs, 7 ATPs, 9, 10 ATPs. Okay. And I have a molecule of fumarate left. So the next enzyme is called fumarase. It takes fumaric acid, which is an alkene, and it puts water into it. Okay, so you could really call that a lyase, couldn't you? See the double bond disappearing. You could really call this a lyase as well. Okay. So now you have a molecule of malic acid because water is put back into that double bond. And then malic dehydrogenase is an oxidoreductase. It has to be able to bind malic acid, NAD, and it oxidizes malic acid into oxaloacetate. So notice that that secondary alcohol there is now oxidized into a ketone. And then I make another NAD, which is worth 3 more ATPs. So I regenerated oxaloacetate. I brought in 2 carbons, lost 2 carbons, right? Now we're carbon neutral. I made 3 NADHs, right? 1 FADH2 and 1 GTP. And I gave off 2 carbon dioxides. I had to put that 2 there because the thing I stole from was wrong. So here's 1 turn of the cycle. Okay. 1 turn of the cycle. This is in the absence of the electron transport system. Because if you had the electron transport system working, all this and this would be turned into ATP. Okay. So that's 9 ATPs, 10, 11 ATPs, right? So 1 acetyl coenzyme A is giving you 11 ATPs here and 1 there. That's 12 ATPs. So one of the things that I remember when I was an undergraduate, I never took a lower level course like this. I just went right into the high one because they didn't have this, I think, at the time. And I remember being asked the questions like, well, follow the carbon atoms. The carbon that you brought in acetyl coenzyme A, is it the same carbon that's taken off over here? And the answer is no. So then they would ask you like, oh, how many turns do you have to go through before you get those 2 carbons taken off? And I don't really think that's important. You just need to know that you cycle through the carbons, right? And that was kind of the issue when they were trying to figure out the Krebs cycle. So maybe you would label the carbon atoms coming into the Krebs cycle. So maybe these are radio labeled with carbon 11 or something like that. And then you're watching the carbon dioxide come off. It's not carbon 11. It takes a couple of turns before you start seeing carbon 11. So carbon 11 is cycling through the intermediates of the cycle. That made it much more complicated to understand.
I have a question. The enzyme succinate dehydrogenase. Did you say is that a lyase or an oxidoreductase enzyme, or is it both, or can it be both?
It's both. Yeah.
Okay.
Yeah. Yeah, because essentially, you have an enzyme that put a double bond in it. Okay, it put a double bond in it by doing an oxidation, but it still put a double bond in it. So here's some regulatory aspects. So pyruvate carboxylase is inhibited by acetyl coenzyme A. So why would that be important? What if you had a whole bunch of acetyl coenzyme A? What would that mean? That would mean that the cycle isn't moving. The cycle isn't moving, possibly because we already have enough ATP, we already have enough NADH and everything. Then why bring more acetyl coenzyme A's into the Krebs cycle? It's not going to be useful. Pyruvate dehydrogenase is inhibited by ATP, NADH, and acetyl coenzyme A. Why does that make sense? For the same reason. Why would you keep trying to bring this in when we already have all this energy? Citrate synthesis, synthetase. is inhibited by ATP right here. Why is that logical? Again, why do this if you have everything full already? Doesn't make sense. And alpha ketoglutarate dehydrogenase is also inhibited by things that represent high levels of energy. Anyway, so here's the overall reaction for a single acetyl coenzyme A before the electron transport system or without oxygen, right? Okay. And here it is after the electron transport system or with oxygen. So you're just simply making all these ATPs, right? So step by step, here's where we're making the energy. So we could say that one acetyl coenzyme A is worth 12 ATPs. So how much energy is produced in complete catabolism of one glucose molecule all the way to carbon dioxide? So glycolysis in many tissues gives you 8 ATPs because many tissues use the malate aspartate shuttle. And then you get 6 ATPs from there. You get 2 acetyl coenzyme A's, so that's 24 ATP there. You add that up. So in heart and liver tissue, because of the malate aspartate shuttle, one glucose is worth 38 ATPs. But in muscle and nerve tissue, only 36 ATPs because you use a different shuttle to get NADH into the mitochondria. Okay. So here's an interesting analysis. A hydrolysis of 1 ATP gives you about 7.3 kilocalories. Okay. So the energy that you get from oxidizing a glucose molecule would be then 38 ATPs times 7.3 kilocalories per mole from an ATP, giving you about that many kilocalories per mole of glucose. But if I burn it in a calorimeter, with very careful controls, I get way more energy. So we assume that when you burn it in a calorimeter in a carefully controlled situation, that's the amount of energy that you can get from a glucose molecule. So the biological efficiency is only about 44%. So you might be thinking that's not very great, but a car is really only about 3% efficient. A hydrogen fuel cell is only about 13% efficient. So here's some metabolic energy calculations. And these are important if you're a physiologist or nutritionist studying metabolism. So glucose can be either 36, 38 ATPs. Let's see how all this works right here. So going back to Krebs cycle and oxaloacetate right here, you need this for the Krebs cycle to be able to keep going. So the cell has to have ways to always have oxaloacetate. So here's one way. There's an enzyme, phosphoenolpyruvate carboxykinase. It takes PEP, puts on a CO2, and it basically makes oxaloacetate at the same time as making GTP. High energy molecule, high energy molecule. So another one is using pyruvate decarboxylase. So it can also produce, at the expense of ATP, oxaloacetate. And there's a couple of amino acids that when you deaminate them, you can get oxaloacetate. So here's aspartic acid, here's asparagine. Both of those can end up giving you oxaloacetate. So, any questions so far? You have all this memorized, right? So you can see here that my goal has never been to make people memorize all this stuff. It's only to help them appreciate how it's logical and how it can be used to figure things out, including diseases.
I think the biological efficiency is really interesting because when companies put their nutrition facts on labels, do they take that into account, or is that just calorimeter calories?
Those are calorimeter calories. So for example, they take a gram of fat and put it in a calorimeter, and that gives them 9 calories per gram, right? And carbohydrate and protein gives about 4 calories per gram. So if you go on a food label and you look at the number of calories, and then you can use that information to actually confirm, oh, this food has so many grams of fat, and then you multiply it by 9 calories per gram. You see what I'm getting? And you add all that up. That's the number of calories that are in that food if you burnt it in a calorimeter.
But we're not getting all of those calories.
No, you're not. Yeah. Yeah, because, you could essentially be like my granddaughter, who the other day I took her to the mall. She's 4, and she's wearing a mask. It was really cute until she got in the car and took the mask off, and there was all that chocolate wiped all over the inside of her mask. Yeah, we don't get 100% efficiency. So this is really what I did my PhD on, the transhydrogenase. So NADH is primarily used to reduce the mitochondrial electron transport system to get ATP. But NADPH is primarily used to provide reducing equivalents for biosynthesis. So when I started my research, this was not known. They thought that the transhydrogenase was to take NADPH and turn it into NADH so you could make energy from it. So I had to refute a lot of publications and make a lot of people think differently. So the transhydrogenase is a large proton translocating integral protein in the inner mitochondrial membrane. It's a reversible enzyme, so it can do either. Remember, enzymes, all they do is get to equilibrium faster, but then it's still subject to Le Chatelier's principle. So if you have a whole bunch of NADPH and you're not going to do biosynthesis, you need energy, you can convert it into NADH that goes into electron transport system. But if you have a lot of NADH, you don't need any more. You can regenerate NAD by making it NADPH, which then can be used for biosynthesis. So there's still some controversy there. But anyway, the role of the transhydrogenase in the maintenance of the glutathione is not even controversial. So glutathione is a tripeptide that's necessary in quenching reactive oxygen species, such as the hydroxy radical. So upon reduction of a reactive oxygen species, glutathione is converted into glutathione disulfide. This is the oxidized form, right? Now you got to get it turned back into glutathione using glutathione reductase, which uses NADPH. So here's the quenching of a radical. So then two of those radicals get together to make the disulfide. So now the disulfide has to be reduced back into glutathione, and it uses NADPH to do that. So when I was doing my research in the 80s, this was not even known, but it became another important reason for the production of NADPH by the transhydrogenase. So it's interesting how amino acids essentially have multiple purposes, don't they? The pentose phosphate pathway is also called a shunt. And what you're doing there in a shunt is you're shunting reducing equivalents or sugars away from one pathway into another pathway. But it shunts glucose away from catabolic pathways into anabolic pathways. So here's how it does that. So here's glucose 6-phosphate. It's going to be oxidized into 6-phosphogluconolactone. And then another enzyme turns it into 6-phosphogluconate. And then notice that you're reoxidizing it again and decarboxylating it, and you end up getting ribose 5-phosphate. So then you have some other reactions that can take ribose 5-phosphate, and that's the ribose. That's the derivation of ribose, and the deoxyribose is made for nucleic acid synthesis. Okay. So we'll see later on that the NADPH is used in biosynthesis, especially for lipids, glutathione reductase, right, that we talked about up above. So it's important. So now we'll switch our emphasis into lipid metabolism. So does anybody have any questions about carbohydrate metabolism before we go on? So notice that this is a biosynthetic pathway, isn't it? Okay, well, what happens to lipids? You eat a lipid and you instantly get fat, right? Well, it takes a while. Even getting fat is complicated. So here's your triacylglycerol. You have lipase. It's either going to be in your intestines or in your fat cells. And it breaks it down into glycerol and into fatty acids. So what happens to glycerol? Well, it's converted into glyceraldehyde 3-phosphate, and it can enter glycolysis to give you energy. Okay. Notice that the reaction uses an ATP, but it also makes an NADH. So this process right here is giving you 2 equivalents of ATP right there. Then what happens to the fatty acids? Okay, they're go through what's called beta oxidation. And you get acetyl coenzyme A that goes where again? Yeah, goes into the Krebs cycle. Beta oxidation is in the matrix of the mitochondria. So is the Krebs cycle. Well, what about these guys? Yeah, they go into the electron transport system, which is right there. So beta oxidation is really like in two distinct steps. The first part is activation. So that results in getting the fatty acid into the mitochondrial matrix. So the fatty acid is condensed with coenzyme A at the expense of 2 ATPs. So the fatty acid, and here's ATP and coenzyme A, is turned into the acetyl coenzyme A, and you break that bond to make AMP, but then later on, pyrophosphate is broken down into 2 phosphates. So now you have what's called an activated fatty acid. So that fatty acid, acyl, because this is the acyl group now, right? In this thioester here. That's transported into the matrix using an amino acid called carnitine. So carnitine, here's the structure of carnitine. So you can see it's an amino and it's an acid. It's a quaternary amine right here. And so where does it bind to the acid? Excuse me, the fatty acid. Right here. So this is an acyl bond, forms an ester linkage between the carnitine alcohol and fatty acid carboxylic acid, acyl group. So then that's combined over here, and now it can be transported through a special transport protein into the matrix. Then the carnitine is released. They can go back again to get another one. But now you have the acyl coenzyme A inside the matrix. Then degradation can happen. And because I don't want to emphasize memorization, I want you to see the big picture. Degradation includes about 4 steps. And so what happens first? Beta oxidation to form a beta double bond and 1 FADH2, then beta hydration to form a beta alcohol, then beta oxidation of that alcohol to give you a beta ketone and another NADH, then beta cleavage to give you another acetyl coenzyme A, and you keep going until the fatty acid is gone. If there were unsaturated bonds in the acyl group, first thing is this isomerized into trans, and then it's hydrogenated. Okay, so it's only hydrogenated fatty acids that go into this machine. What if you have an odd number of carbons? The last oxidation will leave propanoyl coenzyme A with 3 carbons. So then you have to make it into an acetyl coenzyme A by removing that CO2. Okay. So I had to go put this on a separate page because it's so big. So let's see, here's the fatty acid, here's the coenzyme A, so you put it on here. Now you have your activated coenzyme, acyl coenzyme A in the matrix. So here's the first step, oxidation. Well, what's going on? Here's your alpha carbon, here's your beta carbon. You made an alpha beta double bond in the fatty acid. Then the next step, you put water into it. You saturate that double bond. And so it's done in such a way, the enzyme makes it go to the beta carbon, not to the alpha carbon. So now you can see it's alpha beta. Everything's at the beta position here. So when you oxidize that alcohol, you get a beta keto group. And then look at where the cleavage happens. Remember, you're trying to get acetyl coenzyme A, right? You're cleaving it right there at the beta position. So you've shortened it by 2 carbons because those 2 carbons now go off as acetyl coenzyme A. Then you just keep recycling through here until you're done. So activation requires 2 ATPs. So one turn of the cycle gives you an FADH2, an NADH, and an acetyl coenzyme A. And I think you know where we get these energy equivalencies. Then at the end, you end up with one more acetyl coenzyme A, so that's another 12 ATPs. So if we take this fatty acid, and we can figure out how much ATP can be made from the complete oxidation of that fatty acid. Okay, well, where am I going to cut it? I'm going to cut it every 2 carbons because I'm going to make acetyl coenzyme A. So I'm going to cut it here and here and here and here. And I find out that I'm going to get 1, 2, 3, 4, 5, 6, 7, 8 cuts. Every cut gives me 8 FADH2s, 8 NADHs, right? But when I make 8 cuts, I end up with 9 pieces. And every piece is an acetyl coenzyme A, which is worth 12 ATPs. So I can see that most of the energy coming from a fatty acid comes from the acetyl-CoA going into the Krebs cycle. Altogether, I get 146 ATPs from that one molecule. And there's a reason why I stopped it right here. The reason will be apparent when we look at the synthesis of fatty acids, because we're going to start with using acetyl coenzyme A. It's our starting point. Yeah. So anabolism, or lipogenesis. So lipogenesis occurs in the cytoplasm, whereas beta oxidation occurs in the matrix. So there's a very large complex. It's called the fatty acid synthetase complex. Several steps, and anyway, this enzyme right here, we'll be talking about it a little bit later on. Anyway, so let's look at what we have to do. So we take an acetyl coenzyme A, and we have to replace the coenzyme A with this acyl carrying protein, which is also a thiol. And it takes an ATP to do that. So we now have the acyl, the 2 carbons, on what's called the acyl carrying protein. So then it takes the acetyl coenzyme A, with 2 carbons, this enzyme right here, takes a CO2 from the vicinity, right? This dissolved in any part of the solution, but it's in the cytoplasm, so in the cytoplasm, and uses another ATP to make malonyl coenzyme A. That's 3 carbons. So those 2 steps have to happen at the expense of energy. And then we combine the acetyl acyl carrying protein with the malonyl coenzyme A. So this is 2 carbons, that's 3 carbons. We need 2 NADPHs, which is worth 6 ATPs essentially. And then we make a 4-carbon fatty acid, giving off 1 CO2, and that's the beginning of our growing fat. It's got an even number of carbons. Even, odd. Remove that, now it's an even number. And we simply repeat this process until we're done. And finally, an enzyme hydrolyzes the last fatty acyl coenzyme A from the acyl carrier protein to release the fatty acid. Regenerate the acyl carrying protein. We generated the fatty acid. So how much, notice also the NADPH because it's biosynthesis, right? General theme here. So how much ATP was required to synthesize octadecanoic acid, starting with acetyl coenzyme A? Well, we had to do this step right here that took an ATP. Then every time we add 2 carbons to the growing fatty acid chain, we have to use 1 more ATP. Okay. So we had to add 8 to make 16, 17, 18 carbons, right? And then every synthetic step takes 2 NADPHs. So we add all that up, it takes 57 ATPs to synthesize octadecanoic acid, starting with acetyl coenzyme A. Now, if we go back to the catabolism of this 18 carbon fatty acid, well, we're only getting 38 ATPs out of here. But it takes 57 to synthesize it. What does that mean? Well, again, it's not efficient. So the process is really only about 67% efficient. The energy is lost due to entropy, which is randomness, which is heat. It's lost because of energies of activation, and really diffusion as well. Let me see what I did here. Okay, yeah, this is this real quick. So what about synthesis of phospholipids and cholesterol? So this is how you make glycerol 1-phosphate, which would be this right here. And then you have enzymes that combine, the coenzyme A fatty acids. And then here's how you get your, your phospholipid, right? That's a phospholipid. If it's just a regular lipid, you simply use glycerol, not glycerol phosphate. Okay. So here's kind of the mechanism: glycerol, the glycerol phosphate. Here's the hydroxyacetone phosphate, can be converted into this, eventually into that, and into that. Then here's your triacylglycerol. Here's your inositol phosphates, right? So there's just ways to do that, which you don't really have to know that much of. And what about cholesterol? Because you make about 1 gram of it every day in your liver. Well, one way it synthesizes from acetyl coenzyme A. So acetyl coenzyme A can be converted into cholesterol. The enzymes that do that can be inhibited by statins, right? So here's the 2 major enzymes that are inhibited by those statin drugs. And what they are is simply competitive inhibitors to acetyl coenzyme A. You can see the process. And sometimes you have enzymes that do a ring closing, right? And later on, cholesterol is converted into steroid hormones, vitamin D, bile acids, right? When you have too much of this in the environment, it can start going on to proteins and ruining proteins. One of the major, I learned this from some Japanese colleagues. One of the major components of shark liver oil, of which several people I know say, oh, I take that because it's good for your this and that and everything. And it's like, shark liver oil is almost entirely squalene. So I don't know if you should be drinking that stuff or eating that stuff when it's one of the precursors to forming cholesterol. Anyway, so let's stop there, and then when we come back tomorrow, we'll talk about some regulation of these processes. Notice that that enzyme again becomes important because in modern day society, we have huge obesity problems. So people really want to understand obesity. I'm very happy that one of our new faculty members has moved on to the floor with me in the biomed building. This is really her research. So she's using most of my facilities right now. So in my cell culture apparatus, she's growing fat cells, and she comes in every day to see if her fat cells are getting fat. So far, she's been disappointed. They're not fat enough. So does anybody have any questions at all? So don't forget you have your exam to do today if you didn't do it. And let's see. If you do end up having questions, just please email me. And yeah, I will talk to you tomorrow then. Have a great day then.
Thank you.
Take care.